Welcome to the Deep Dive, where we plunge into the heart of fascinating topics to bring you those crucial nuggets of knowledge. Today, we're embarking on a deep dive into something truly remarkable, a part of genetics that was, well, for a long time, surprisingly overlooked. We're talking with the unsung heroes completely reshaping our understanding of how life works. Non-coding RNAs, or ncRNAs.
That's right.
For decades, the spotlight in genetics shone almost exclusively on DNA and proteins. But just imagine if, 80% of the genetic transcription happening at a typical human cell was largely ignored, not just in research, but even in education.
Yeah, it really was a huge historical bias against RNA. It's quite astonishing when you think about it. For so long, the story was, all about protein encoding genes. DNA makes mRNA, mRNA makes proteins. Simple. But what we're now realizing, and it's a big realization, is that the script was far more complex. And these ncRNAs, they're not just supporting characters. Often they're actually directing the whole show.
Exactly. And today, we're going to dive deep into that richer, more accurate story. You're about to discover that ncRNAs, these fascinating RNA molecules that don't encode proteins, are performing just a spectacular array of functions. In fact, they're often more abundant than even messenger RNAs in most cells, playing critical roles in, well, everything from how our DNA is copied and packaged to how proteins are made and even how our genome defends itself. It's huge.
Absolutely. Our mission for this deep dive is to really distill the essence of what ncRNAs are. We'll be using Chapter 17 of Brooker's Genetics, Analysis and Principles, 7th edition, as our guide. We'll explore their diverse roles, the groundbreaking experiments that unveiled their secrets, and their surprising, often profound connection to human diseases.
Yeah, get ready for some serious aha moments that might fundamentally change how you view the inner workings of your cells. Okay, let's unpack this, starting with the basics. What exactly are non-coding RNAs? We've said they don't encode polypeptides, but put that in perspective.
Right. So humans have roughly, 22,000 protein encoding genes, give or take. When it comes to ncRNA genes, the estimates range from thousands to potentially tens of thousands.
Wow, that's a massive difference. A vast universe of molecules we're still figuring out.
An incredible diversity, yeah. And the real superpower of ncRNAs, the reason they can do so much, it really lies in their ability to bind to various other molecules.
Okay.
They can bind to DNA or other RNA through complementary base pairing, like A with U, G with C, that's familiar. But they also bind very specifically to proteins, often using these unique 3D shapes like stem loop structures.
And these intricate folds.
Exactly. And they can even bind to small molecules, too.
So it's not just a simple interaction. Some ncRNAs are like molecular multi-tools.
Yeah, that's a great way to put it. Yeah, multi-tools, they can have multiple binding sites, letting them bring together several different molecules, often proteins, forming complex structures.
Like orchestrating things.
Precisely. Orchestrating interactions. And this incredible versatility allows ncRNAs to fulfill, well, the textbook outlines 6 general functions. It's like their toolkit. First, they can act as a scaffold. Imagine a molecular workbench holding multiple components together, maybe different proteins, in just the right way to form a complex.
Like building cellular machinery.
Exactly, an assembly line. Second, building on that, they can act as a guide. Because an ncRNA can bind a protein and maybe a specific spot on DNA, it can literally guide that protein right where it needs to go.
Like a biological GPS. Precision targeting.
You got it. Third, crucial function. Alteration of protein function or stability. When an ncRNA binds to a protein, it can actually change that protein's 3D shape.
Oh, interesting.
And that can affect its catalytic activity, how well it binds other things, or even how long the protein lasts in the cell before it gets degraded.
Okay, this next one is truly fascinating. Some ncRNAs function as ribozymes.
Yes, RNA molecules with catalytic activity, like protein enzymes, but made of RNA. It was a mind-blowing discovery.
A classic example being RNAs.
Right, RNAs, it has an RNA component that acts as the enzyme to cleave tRNA molecules, trimming them to the right size. It's amazing.
Okay, fifth function.
They can act as a blocker, literally just getting in the way. For instance, an antisense RNA that's an ncRNA complementary to a messenger RNA if it binds right over the start codon.
It physically stops translation from starting, like a roadblock.
Exactly, a molecular stop sign. And finally, number 6, they can act as a decoy. This is quite clever. A decoy ncRNA binds to other ncRNAs, basically taking them out of action.
Soaking them up almost.
Yeah, sequestering them. For example, a decoy might grab a microRNA that normally inhibits mRNA translation. By luring away the microRNA, the decoy lets the mRNA get translated.
Overriding the inhibition. Very neat.
Now, just quickly, we often categorize them by length. You have long non-coding RNAs, ncRNAs, longer than 200 nucleotides, and then small regulatory RNAs, shorter than 200 nucleotides. MicroRNAs, or mRNAs, are a key example here, usually just 20-25 nucleotides long.
Tiny but mighty. Now, all this versatility of RNA, it leads us to one of the most profound ideas about the origins of life. The RNA world hypothesis.
Ah, yes, the RNA world. The idea is that before cells as we know them, you had these precursors called protobions, maybe simple aggregates of molecules with some kind of boundary.
Like a primitive cell membrane.
Perhaps. And in this hypothetical RNA world, RNA, not DNA or proteins, was the star player. It's proposed that RNA performed all three key functions: storing genetic info, self-replicating using ribozyme activity, and carrying out various catalytic functions.
Wow. So RNA did everything. Information, replication, catalysis, the original multitasker.
That's the hypothesis. It suggests RNA was the primary macromolecule.
So what drove the shift to our modern DNA and our protein world? What were the advantages?
So specialization offered big advantages. DNA is much more stable for long-term information storage, chemically speaking. And proteins, with their 20 amino acid building blocks compared to RNA's 4 bases, have vastly superior catalytic abilities and structural diversity.
So a more stable library with DNA and more powerful tools with proteins. Makes sense.
It's an elegant evolutionary leap. But RNA didn't disappear, obviously. It's still absolutely central, especially in protein synthesis. Think mRNA, tRNA, rRNA.
And even the ribosome itself, the protein-making machine, its catalytic core for forming peptide bonds is actually rRNA.
Exactly. A fantastic echo of that ancient RNA catalytic power.
Okay, so we have a grasp of what ncRNAs are and their amazing toolkit. Let's dig into how they work in specific cases. Let's start with influencing chromatin structure and transcription. The case study here is HOTAIR, right? Hoax transcript antisense intergenic RNA.
Yes, HOTAIR. It's an lncRNA, about 2.2 kilobases long, and it's a master regulator of gene repression. It silences genes by altering chromatin.
And its mechanism is pretty sophisticated, involving both scaffolding and guiding.
It really is. First, it acts as a scaffold. It physically binds to 2 different histone-modifying enzyme complexes: PRC2, polycomb repressive complex 2, and LSD1, lysine-specific demethylase 1. PRC2 binds near the 5' end of HOTAIR, LSD1 near the 3' end.
So it gathers these 2 modification machines. Then how does it target them?
That's the guide function. HOTAIR has sequences that allow it to bind to specific GA-rich regions on the DNA, near target genes, like certain HOXD genes. This binding guides the PRC2 and LSD1 complexes right to that specific genetic location.
And once they're there, they modify the histones.
Yeah.
Those proteins DNA wraps around.
Exactly. PRC2 adds methyl groups specifically. It trimethylates lysine 27 on histone H3. LSD1 does the opposite. For another site, it demethylates lysine 4 on histone H3.
And these histone marks change how the DNA is packaged.
Correct. These marks are signals. They can directly inhibit RNA polymerase, or they attract other proteins that compact the chromatin. The result is a closed chromatin conformation, which effectively silences the target gene. No transcription.
And the significance when HOTAIR expression goes wrong.
It's bad news. Abnormal HOTAIR expression is linked to many human diseases, especially cancers like breast, lung, and colorectal cancer. It often acts like an oncogene, promoting tumor growth and metastasis.
Wow. Okay, so from silencing genes by changing chromatin, let's switch to how ncRNAs affect things after transcription translation, mRNA degradation, even modifying other RNAs. This brings us to a really huge discovery: RNA interference, or RNAi.
Oh, absolutely monumental. Nobel Prize-winning work by Andrew Fire and Craig Mello in 1998.
Yeah.
It started, actually, with some puzzling observations. Researchers knew injecting antisense RNA could inhibit gene expression. It binds the mRNA, blocks it. Makes sense.
But things got weird, right?
Yeah. They noticed that injecting sense RNA, the strand, with the same sequence as the mRNA, sometimes also inhibited the gene. That didn't make sense.
And the silencing effect seemed weirdly potent and long-lasting sometimes.
Exactly. The pieces didn't fit. So Fire and Mello decided to systematically investigate this in the nematode worm C. elegans. They targeted an mRNA called MEX3. They injected single-stranded MEX3 antisense RNA into the worms.
Okay.
Or they injected double-stranded RNA made by mixing the sense and antisense strands together.
And that was the key experiment.
That was the bombshell. The double-stranded RNA was dramatically far more potent at silencing MEX3 than the antisense RNA alone. In fact, the double-stranded RNA often led to complete degradation of the MEX3 mRNA. gone.
Whoa.
They called this phenomenon RNA interference, RNAi. It was like discovering a cellular off switch for specific genes triggered by double-stranded RNA.
A total paradigm shift. So how does it actually work? What are the molecular players?
The key players in RNAi are two types of small regulatory RNAs, microRNAs, mRNAs, and small interfering RNAs.
What's the difference between them?
Well, mRNAs are typically endogenous and coded by our own genes. They usually have only partial complementarity to their target mRNAs, often targeting multiple different mRNAs. Think of them as fine-tuners. Humans have maybe around 2,000 mRNAs, potentially regulating something like 60% of our protein coding genes. Huge impact.
Okay, and snoRNAs.
snoRNAs are often exogenous, coming from outside sources, like viruses, or researchers can introduce them experimentally. They usually have a perfect or near-perfect match to a single target mRNA, making them very specific. This makes them great for defense, especially in plants against viruses.
Got it. So you have these precursor RNAs. How do they become active?
Right, they need processing. mRNAs start as primRNAs in the nucleus, get processed to pre-mRNAs, then exported to the cytosol. Pre-sRNAs, often from longer double-stranded RNA, usually start in the cytosol. In the cytosol, both types are chopped up by an enzyme called dicer into short double-stranded fragments about 20-25 base pairs long.
Dicer. Okay, then what happens to these short pieces?
These short double-stranded RNAs then associate with proteins to form the RNA-induced silencing complex, or RISC. Inside RISC, one RNA strand is usually degraded. The remaining single strand, now an mRNA or sRNA, acts as the guide.
Guiding RISC to the target mRNA.
Exactly. The guide RNA uses complementary base pairing to find and bind specific mRNAs.
And once RISC binds the mRNA, what does it do? Silence it how?
There are a few outcomes. If the match is partial, like often with mRNAs, RISC typically just inhibits translation. It sits on the mRNA and prevents the ribosome from making protein. Sometimes the mRNA might get temporarily stored in a P body, a processing body.
But if the match is perfect, like with sRNAs,
then RISC usually directs the degradation of the mRNA. An enzyme within RISC, often an argonaute protein, cuts the mRNA, destroying it. Complete silencing.
so it's a really versatile system for gene regulation and defense.
Incredibly important, yes. Fundamental regulation and a key antiviral defense, particularly in plants.
Okay, moving beyond controlling mRNAs, ncRNAs also modify other RNAs, like ribosomal RNAs. This is where snoRNAs come in. Small nucleolar RNAs.
That's right. snoRNAs hang out mainly in the nucleolus, which is the ribosome factory of the cell. Their main job is to guide the chemical modification of ribosomal RNAs. Two common mods are methylation of the ribose sugar and converting uracil to pseudouracil.
And why modify the RNAs?
These modifications seem to fine-tune the RNA structure, which is crucial for the ribosome to work properly, things like folding correctly and catalyzing peptide bond formation accurately during translation.
So how do snoRNAs guide these modifications? Is it that scaffold and guide thing again?
Exactly. It's a beautiful example. Each snoRNA acts as a scaffold for specific proteins, forming a complex called a snoRNP, small nucleolar ribonucleoprotein. There are different types, like CD-box snoRNAs that bind methylating enzymes, and HACI-box snoRNAs that bind pseudouridylation enzymes.
So they package the right enzyme, and they guide it.
Correct. The snoRNA part of the snoRNP has antisense sequences that act as a guide, binding specifically to the target site on the RNA. Once positioned, the snoRNP's enzyme catalyzes the modification. Highly precise targeting.
Incredible precision. Okay, let's switch tracks again. What about getting proteins to the right place in the cell? There's an ncRNA involved there too.
Absolutely. The signal recognition particle, or SRP, this is a crucial RNA protein complex. Its job is basically protein targeting, acting like a molecular escort service. In eukaryotes, it guides proteins destined for secretion or insertion into membranes to the endoplasmic reticulum, the ER.
So it reads the protein's address label.
Pretty much. In eukaryotes, the SOP is made of one ncRNA molecule and 6 different proteins.
How does it work? Let's walk through the eukaryotic process.
Okay, so a ribosome starts translating an mRNA in the cytosol. If the protein being made has an ER signal sequence, that address label sticking out.
SRP spots it.
Yes, SRP recognizes and binds to that signal sequence and the ribosome. This binding actually pauses translation temporarily, halts production.
Buys some time. Then what?
Then the whole complex SRP ribosome pause polypeptide docks onto an SRP receptor protein embedded in the ER membrane. This docking needs energy, requires GTP binding by both SRP and its receptor.
Okay, it's docked. How does it hand off the protein?
Through GTP hydrolysis. Proteins within both the SRP and the receptor burn their GTP. This energy release causes the SRP to detach from the receptor and the ribosome. Translation then resumes, and the polypeptide chain is threaded through a channel right into the ER lumen.
And the SRP RNA's role in all this.
It's critical. First, it acts as the scaffold, holding those 6 proteins together correctly. But maybe even more importantly, the SRP RNA actually stimulates the GTPase activity of the proteins in both the SRP and the receptor. This stimulation is essential for the timely release of SRP, letting translation restart.
Very cool. Okay, let's shift to defense mechanisms, particularly in prokaryotes. The CRISPR-Cas system, hugely famous now, of course.
Right, CRISPR-Cas, found in many bacteria and most archaea. It's an adaptive immune system against foreign invaders like bacteriophages, viruses that infect bacteria, plasmids, things like that.
Adaptive meaning it learns from past infections.
Exactly. The cell has to encounter the invader first to build immunity. It works in 3 phases: adaptation, expression, and interference.
Let's break that down. Adaptation phase.
So after a phage infects the bacterium, special Cas proteins, Cas1 and Cas2, recognize the foreign phage DNA. They chop it up. Then they take a small piece of that phage DNA, called a spacer, and integrate it into a specific region of the bacterial chromosome called the CRISPR array.
Like adding a mugshot to a most wanted list.
That's a perfect analogy. It creates a genetic memory of the invader, which is then inherited by the bacterium's descendants.
Okay, so the cell is primed. What happens if the same phage attacks again? Expression phase.
Right. Now, the CRISPR array, containing those stored spacer memories, gets transcribed into a long ncRNA called pre-chRNA. At the same time, another nearby gene, TRAC, makes TREC-RNA. The TRAC-RNA base pairs with the pre-chRNA. This duplex is then processed into mature, shorter cRNAs, each still bound to a piece of TREC-RNA. This srRNA-TREC-RNA complex then recruits the key enzyme, the Cas9 protein.
Got it. So now you have this complex armed with a memory, the cRNA spacer, and a weapon, Cas9, interference phase.
Now it's ready for action. This whole complex, TRAC-RNA, shRNA, Cas9, acts as a guide. It patrols the cell. If it encounters invading DNA that matches the spacer sequence in its cRNA, it binds tightly.
Match found.
Match found. And then Cas9, which is an endonuclease, acts like molecular scissors. It makes double-strand breaks in the invading phage DNA,
Lethal cuts.
inactivating the virus.
Exactly. It neutralizes the threat.
And of course, the incredible part is how scientists repurpose this natural bacterial system into the CRISPR-Cas9 gene editing tool. Amazing story.
Truly revolutionary. Another deep dive, perhaps.
Definitely. Now, staying with genome defense, but switching to animals. PIWI interacting RNAs or pRNAs?
Yes, pRNAs. These associate with PIWI proteins, and they primarily function in germline cells, the ones making sperm and eggs. Their main job is to fight transposable elements, or TEs. Those are the jumping genes that can move around the genome and potentially cause damage or mutations.
So pRNAs are guarding the genetic integrity passed to the next generation. Precisely. Keeping the T's in check. Pre-pRNAs are processed into mature pRNAs, about 24-31 nucleotides long, complementary to tRNA sequences. These pRNAs team up with PIWI proteins to form complexes that silence TEs in 2 main ways.
Okay, what are they?
First, there's the pRNA-induced transcriptional silencing complex, or PRITs. This complex goes into the nucleus, it binds to nascent TE transcripts, RNA being made from a TE, and then recruits machinery to chemically modify the TE's DNA and associated histones, DNA methylation, H3K9 trimethylation.
Shutting down the TE gene itself by changing the chromatin.
Exactly, packing it into silent heterochromatin, stop transcription at the source. The second way involves the pRNA-induced silencing complex, or PRC, which works in the cytosol. This complex binds to mature tRNA transcripts that have already been made. A protein within pRNA, maybe an argonaut like an RNAi, then simply cleaves the tRNA, destroying it.
So transcriptional silencing and post-transcriptional degradation, a double whammy against TEs.
A very robust defense, yes. And pRNAs are incredibly diverse. They're the largest class of ncRNAs known, primarily for TE defense, but they likely regulate some normal genes too.
So after all this controlling genes, modifying RNA, targeting proteins, defending the genome, it's probably no surprise that when ncRNAs go wrong, it can lead to disease.
Absolutely. Abnormal ncRNA expression, maybe from mutations or epigenetic changes, is linked to a huge range of human diseases. It's a major area of current research.
What was the first connection discovered?
The very first ncRNA gene linked to a genetic disorder was actually for RNase MRP, part of a ribozyme complex. Mutations cause cartilage hair hypoplasia, or CHH. Patients have issues like short stature, sparse hair, skeletal problems, immune defects, and higher cancer risk. You showed early on how vital these ncRNAs are.
And the connection to cancer seems particularly strong and well-studied, especially with mRNAs.
Definitely. We see altered mRNA levels in almost all human cancers studied. Some mRNAs act like oncogenes. If they're overexpressed, they drive cancer growth. Others act as tumor suppressors. If their levels are too low, it allows tumors to grow or spread.
Can you give an example of a tumor suppressor mRNA?
A great example is the miR-200 family. Low levels of these mRNAs are linked to aggressive metastatic cancers, bladder, melanoma, stomach, colorectal, and others. miR-200 normally helps keep cells stuck together by maintaining levels of E-cadherin. When miR-200 is low, cells lose E-cadherin, become less sticky, and can undergo this process called EMT epithelial mesenchymal transition, which is often the first step in metastasis.
So maintaining miR-200 helps prevent cancer spread.
That's the idea. And remember HOTAIR, the ncRNA we talked about earlier?
The chromatin modifier.
Yes. High levels of HOTAIR are found in many cancers, like breast and lung cancer, and it acts like an oncogene, correlating strongly with metastasis. And even pRNAs and PIWI proteins are implicated in some testicular cancers. The links are widespread.
Beyond cancer, neurological disorders also seem heavily influenced by ncRNAs.
Hugely. Something like 70% of all known mRNAs are expressed in the brain, many specifically in neurons. So it's not surprising that abnormal mRNA levels are associated with almost every neurological disorder investigated.
Such as?
Well, in Alzheimer's disease, certain mRNAs are thought to regulate the enzyme beta-secretase, affecting the production of those toxic amyloid peptides. In multiple sclerosis, mRNAs seem involved in controlling the neuroinflammation, and even mutations in the mRNA processing machinery, like the enzymes DROSHA or DICER, are linked to diseases like familial ALS, amyotrophic lateral sclerosis, often causing a general decrease in many mRNA levels.
And cardiovascular diseases, too.
Yes, definitely. Abnormal mRNA levels are linked there as well. For example, miR-1 is involved in regulating ion channels critical for heart rhythm, so its dysregulation is linked to arrhythmias. We see specific mRNA signatures in patients with heart failure, and others, like miR-NA, miR-145, miR-143, are implicated in the development of arterial plaques, contributing to atherosclerosis.
Okay, so given this deep involvement in so many major diseases, the big question becomes, can we target ncRNAs therapeutically?
That's the exciting frontier right now. If ncRNAs are causing or driving disease, can we intervene? Can we manipulate them for treatment?
Let's take cancer, where a mRNA is acting like an oncogene is overexpressed. How could you stop it?
The leading strategy is using anti-mRNA oligonucleotides, or AMOs. These are short synthetic nucleic acid strands designed to be complementary to the problematic mRNA. They bind to the target mRNA and either just physically block it from working or sometimes lead to its degradation.
Are there specific types of AMOs being developed?
Yes, researchers are engineering them for better stability and binding. Locked nucleic acids, LNAs, have modified structures that make them bind very tightly. Antagomeres are another type, chemically modified for stability and potent inhibition. Early studies, mainly in mice so far, have shown promise, for instance, using an antagomere against miR-10B to inhibit breast cancer metastasis.
Very promising. What about the flip side? When a mRNA is a tumor suppressor and its levels are too low?
Then you want to restore its function. One approach is mRNA replacement therapy. This might involve using, say, a harmless virus as a vector to deliver the missing mRNA back into the cancer cells. Studies in mouse models, like restoring miR-26A in liver cancer, have shown this can inhibit tumor growth.
Are there other ways to boost mRNA levels?
Potentially. Some research looks at targeting the mRNA processing machinery itself. A drug called enoxacin was found to enhance overall mRNA processing, boosting levels of many mRNAs and inhibiting tumor growth in mice, interestingly, without harming normal cells.
That sounds quite elegant.
Another approach is tackling the root cause. If the mRNA gene itself has been silenced epigenetically, maybe through DNA methylation or histone changes.
Like using drugs to remove those silencing marks?
Exactly. Using DNA demethylating agents or histone deacylase HDAC inhibitors can sometimes reactivate the expression of silenced tumor suppressor mRNAs. And the exciting thing here is that some drugs in these classes are already clinically approved for treating certain cancers, so the potential to repurpose or combine them to restore mRNA function is very real.
What a truly profound journey we've taken today,
Yeah.
seriously. From thinking about life's origins in a hypothetical RNA world,
Right.
all the way to these incredibly sophisticated ways ncRNAs, orchestrate our genes, defend our cells, target proteins, and unfortunately, also play roles in disease.
It really hammers home how molecules once dismissed as, maybe just junk or minor players are actually fundamental.
Absolutely. They're at the forefront of genetics research and increasingly therapeutic development now.
It really just changed your whole perspective on cellular biology. And when you think about that amazing versatility, scaffold, guide, enzyme, blocker, decoy, it makes you wonder, what other fundamental processes might ncRNAs be quietly controlling that we just haven't discovered yet?
That's the big question, isn't it?
Yeah.
And maybe the even bigger one. How might our growing ability to actually manipulate these molecules reshape medicine? Could we tackle diseases that seem intractable today by targeting ncRNAs?
It feels like an incredibly exciting frontier. The potential seems enormous.
A truly limitless horizon of discovery, as you said. Well, thank you for being part of the Deep Dive family today. We sincerely hope this deep dive has given you a fresh and maybe even profound perspective on the incredibly dynamic and often surprising world inside every one of your cells.